So thank you very much. I'd like to introduce the director of the NIEHS and the National Toxicology Program, Dr. Linda Birnbaum.
Well, first of all, let me welcome all of you here. I hope people didn't have as much trouble as I did coming in this morning and getting through security. There's, at least when I drove up, there was a big backup, and I actually, since I was sitting there, I called security and said, you got to do something about this. I still sat. But anyway, I think what we're talking about today and in the whole Halifax project is that the toxicology of the 21st century should not be the toxicology of the 20th century. That we need to move from the kinds of studies and the kinds of analysis we did in the 1960s and the 70s and 80s to what we should be doing as we approach 2020. There have to be changes in the way we do things and the changes in the way that we interpret data. And I think there's a whole movement to do this and moving toxicology into more predictive science. But we also have to think of the issues of dose. And so one of the key issues that we need to think about is low dose. And I will remind every one of you, when you think about low dose or you talk about it, define what you mean by low dose. Low relative to what? Are you talking about low dose meaning general human population exposure or the range of human exposure? Is that what the mean of low dose is? Or is the mean of low dose lower than an RFD? Or lower than an OL? Define what you mean, because I think it's very important. We're certainly seeing lots of effects now occurring both in experimental animal models and in people at general background exposure. To me, that's what I think about as low dose. Another thing that we really have to start addressing, and I've actually been doing some mixtures work for the last 30 years, but I think that we have to be thinking about mixtures. And it's not just chemical mixtures, A lot of the focus today is going to be on having multiple exposure to exogenous xenobiotics. But remember, your nutrition can totally modify your response to chemicals. Your microbiome, your infectious state, I won't call the microbiome infectious, but your infectious agent state, physical agents, all of these things can modify how we respond, and we've got to start finding out ways to deal with it. And NIEHS just did sponsor a workshop last month focusing on mixtures and epidemiological studies. We're looking at the statistical approaches to try to deal with that. So stay tuned for the workshop report to come out of that. I think that'll be very important. And I think another key issue that until now until relatively recently, we haven't fully addressed is the issue of windows of susceptibility. And I often say a good start lasts a lifetime, or the opposite is true as well. So there's this whole relatively new field of developmental origins of health and disease. But the point is what happens to you early in life, maybe even before conception, has impacts on you and maybe on your children and your grandchildren, and in certain cases, maybe even beyond that. And again, these are issues that we're going to have to start dealing with in the 21st century. So I'm really happy that bringing this group of international as well as U.S. experts together are going to really help us deal with some of these underlying concepts and think about it and strategize on it as we go forward in the area of cancer explicitly, but we will explore other areas as well today. So I'm very excited. I'm looking forward to spending the day with you. And I'm in a moment going to invite Margaret Kripke up to speak, but first I just want to introduce her. It's a real honor to get to know Margaret. I've been familiar with her work on UV and cancer for many years. She's really been a leader and maybe really the founder of that whole field. She is currently Professor of Immunology and Executive Vice President and Chief Academic Officer at the University of Texas MD Anderson Hospital. I think I was interested in seeing, Margaret, that your bachelor's and master's were in bacteriology, since my degrees are in micro as well, or microbiology. But her PhD was in immunology at UC Berkeley. And she's just completed 2 terms on the President's Cancer Task Force, where she has been a voice for the role of the environment and genes. And I think we all need to move forward thinking about doing both. Margaret?
Thank you very much. My task this morning is to talk about environmental carcinogenesis setting the stage. And so in about 2008, the President's Cancer Panel, of which I was privileged to be a member, decided to devote its annual report to the White House on environmental carcinogenesis. And the rationale, I must say, I wasn't wildly enthusiastic about this as a subject, but I became convinced that this was the right thing to do because in the 40-year history of the Cancer Panel, they had never looked at environmental carcinogenesis. It simply had never been done. The second rationale was that it seemed to me, as someone who had been a researcher in the field of environmental carcinogenesis, that this was an understudied and underserved area. If you look at the programs for the last 40 years for the things like the American Association for Cancer Research meeting, there's very little subject matter devoted to environmental carcinogenesis. And in terms of impact, the possibilities for prevention in terms of reducing the burden of cancer are much more substantive than our attempts to cure advanced cancer. And so the President's Cancer Panel has the charge of looking broadly across the cancer landscape and trying to make recommendations about things that would really have an impact on the disease across the population. And it seemed to us that prevention was, again, a way to do that. And finally, there was increasing evidence of the importance of environmental carcinogenesis, largely through the work on endocrine disruptors. And there was growing evidence at that time that these were important. They were environmental constituents that may actually be in part responsible for increasing incidence of certain types of cancer. Now, in those days, the dogma of the day was that the majority of cancers were caused by bad genes and lifestyle factors. And it was thought that only about 6% of cancers could be attributed to environmental causes. And of course, as recently as this year, we read that in a highly cited paper from Johns Hopkins, the claim that cancer is simply the result of bad luck. And so there's never been a very strong sentiment that environmental causes were particularly important for a whole variety of reasons, obviously. So during our year of studying this issue, we became convinced otherwise. And for example, it became very clear during our hearings and listening to testimony that although some carcinogens in the environment and in the workplace are highly regulated, the regulations are not always enforced. There are also known cancer-causing agents in our environment that are not regulated. Now, this came as a great surprise to me, who thought that we were regulating all of the important things. And then we learned that tens of thousands of chemicals in our daily use products and on our grocery shelves and whatever have never been tested for carcinogenicity. That was another surprise to me. And then there was, I think, the most compelling evidence that there were continuing increases in the incidence of childhood cancers, which started in the 50s and 60s and continue to this day. Now, this is very hard to explain on the basis of bad luck or bad genes. So I became very interested in this issue, and we authored a report at the end of the year called Environmental Carcinogenesis, and we came to the very unpopular and very controversial conclusion that the true burden of environmentally induced cancer has been grossly underestimated. And we were heavily criticized because we really had no data to support this contention of ours, aside from the logic of what seemed very obvious to us from all of the testimony that we heard. And all we really had was a conviction based on our observations that 6% couldn't be right, and that the true picture of the contribution of environmental chemicals to carcinogenesis had yet to be revealed. So part of the problem was that there were serious obstacles to determining the answer to the question, how important are environmental chemicals in cancer development? And some of these obstacles included the focus on testing one chemical at a time, which is obviously inadequate. Second was the inability to monitor exposure in people over a lifetime. A third, as was mentioned, was the inability with contemporary approaches to deal with mixtures of chemicals that we are all exposed to over our lifetime. And importantly, the focus on chemicals as mutagens and complete carcinogens, even though we've known about the processes of initiation and promotion for over 50 years, somehow that has not translated into the thought process of regulating carcinogenic agents. In 1990, actually, I wrote a paper, a speculative paper, on the ways in which ultraviolet light could contribute to melanoma induction. In those days, it was quite controversial as to whether ultraviolet light induced melanoma or not, because the UV signature mutations, which are so prevalent in non-melanoma skin cancers, mutations in P53 have a UV signature, so it's very clear that UV is related to those cancers. Those types of signature mutations had not been found at that point in melanoma. And so the point of my paper was to argue that although ultraviolet light is a complete carcinogen, it's also a lot of other things. It can be an initiator, which is promoted by chemicals. It can be a promoter of cancers initiated by chemicals. And it could also decrease local immunity in the skin in a way that permitted melanomas to grow. And so there were many ways in which a seeming complete carcinogen could actually participate in the process, and the fact that you didn't find UV signature mutations in melanoma was not an argument that it wasn't contributing to the disease. So although this was a very logical concept, there was really no way to apply it or incorporate it into our thinking about environmental carcinogens until now, and enter the Halifax project. I think this points the way for us to really a new era of environmental carcinogenesis and causes us to rethink what is the definition of a carcinogen. So let's hear about this groundbreaking work that has the potential to really revolutionize the field of environmental carcinogenesis and hopefully provide us with a more realistic answer to the question of what role do environmental chemicals play in cancer development across the population. And so let's now hear from Mr. Leroy Lowe, who is the founder and president of an organization called Getting to Know Cancer, which is responsible for the Halifax project. Leroy.
Thank you, Margaret. Well, good morning, everyone. First of all, thank you, Linda, for bringing us here, and your staff for hosting us. I'd like to special thanks to Ken Cook at EWG for you and your team for making this meeting happen. David Carpenter is not able to be with us, but I wanted to thank him because he was involved in helping connect us with the NIEHS initially, who was the sponsor of the Halifax project. Michael Gilbertson, my co-founder, who's here in the audience. The project researchers who are here from the Halifax project, welcome, and to all other guests in the room, nice to see you, and thank you for having us. I have an unusual task this morning in setting the stage. I did a look at the list. Many of you won't know my background, but I've no formal training in cancer biology. I've been doing this for about 15 years now. I'll tell you a little bit about the story. That's part of what we're going to do to sort of set the stage for the Halifax project. But I want to talk a little bit about the biology of cancer and a little bit about environmental toxicology. And I'm not going to go very deep in either, but I was looking at the list of people in the room, and I was thinking I'm probably the least qualified person here to do this talk. But what I want to make sure is that everybody's on the same page today. I want everybody to, the people that are in toxicology and environmental health, really understand at a high level what we are really interested in for the biology of cancer, and for those who are cancer biologists, what it is about toxicology that has us focused. And I won't do either justice if you're in either camp, but please bear with me as I sort of give you the broad strokes, because I think it will be very helpful in making sure that this afternoon's discussions are fruitful. So I started, I'm an autodidact, which, as I said, means I'm the least qualified person here. I had to look it up when somebody told me I was an autodidact. I spent 8 years studying cancer research at a time when the Hallmarks of Cancer framework hadn't been developed, or it was developed while I was studying papers. I was reading peer-reviewed literature, and I was reading broadly, and I was reading across all the domains that we know of now as domains, subdisciplines within cancer biology, and I really was lost for a long time. I read paper after paper after paper, going back to relearning my biology. And anyway, in 2000, when the first Hallmarks of Cancer paper came out, for me, it represented a bit of an orienting document. It helped me start to figure out of this broad field of research how I could sort of see. Then when the second paper came out in 2011, and I had called the authors of the paper, Bob Weinstein in particular, I spoke to him directly and said, you know, I like your paper, but there's a bunch of things that I don't see in there that I'm reading elsewhere. Where does that fit? And he said, oh, we're coming out with another paper. And he did come out in 2011 with the second paper. And collectively, it does a nice job of capturing the field. It's not a perfect paper, and people have emailed me and said, you know, it's not a theory of carcinogenesis, for one. I'll mention that. It's not a theory of how cancer gets started and evolves. It's a description of what happens in cancer. We liked it, I'll tell you why I liked it initially. Why I liked it initially was it helped me understand the field. And I'm just going to go very high level through the hallmarks, because I don't know how many of you, if I said take a blank sheet of paper and tell me what the hallmarks are and what they mean, I don't know how many of you could do that, but it's taken me quite a while to get my head around it, and I want to give it to you very quickly. Catherine, who's a recent graduate, just did it in a single semester course. I mean, we've spent 30 years getting to the point where we have all this research, and the Hallmarks of Cancer papers are now, you know, highly cited as it helps us. You can take it in a half semester at university. So I mean, it's at a level that we're being rewarded for all that detailed bench work that was done, and we can now understand it at a level that can help us make strategic decisions, which I'm hoping we can do here today. So we'll start with what is not a hallmark, but stability is referred to as an enabling characteristic in the papers by Hanahan and Weinberg. But we know that tumors are genetically unstable, and an immortalized cell will produce clones of itself that are not exactly the same. And it's that heterogeneity that gets produced in producing clones that aren't the same that makes cancer so hard to treat, because they're not, the subpopulations that are produced are also immortalized, but they're not all the same genetics. And for that reason, genetic instability is important, and it's seen across all cancer types. It's a commonality that's shared. Cancer cells have sustained proliferative signaling. They've managed some way to find a way to keep growth signals going so that they keep making copies of themselves. And there are tumor suppressors that represent anti-growth signaling, if you will, ways to stop a cell. P53, for example, is a tumor suppressor. It's quite famous, known as the guardian of the genome. If there's damage in a cell, it can stop the cell cycle from going further and make sure that repairs are effected to the damage that's been done. If things are fine, it can allow the cell to proceed and replicate. If things are damaged irreparably, then it can send the cell to senescence, or a form of dormancy, or to program cell death and for self-destruction. And so that's really important. And these tumor suppressors have been bypassed or are somehow bypassed in cancers that we see. There is a resistance to program cell death. Cells that are supposed to self-destruct don't, and they should. That machinery, and literally it is machinery within the cell, has been bypassed. There's something called replicative immortality. These immortalized cells can make copies of themselves for a much longer period of time than they should be able to. There's machinery that allows a cell to keep track of how many times it's made copies of itself, and there's a limit. And that limit has been bypassed, and the machinery has been disrupted in a manner that allows it to be bypassed. The metabolism within the cell has changed. The normal cellular metabolism in a hypoxic environment with all these cells cloistered together becomes dysregulated, and that metabolism shift changes things and enhances the genetic instability within the cell itself. The immune system has been somehow bypassed. The immune system the cell has its own ability to say, hey, I'm in trouble. And there's an immune system, natural killer cells, dendritic cells, macrophage, T cells, all of which have a role to play in shutting cancer cells down. And they have somehow not managed to stop the tumor from developing. And then we have angiogenesis, this process of having blood vessels provide fuel, if you will, oxygen and blood to the cells in and around the tumor in the microenvironment. And that angiogenesis is occurring in a very dysregulated manner. It's a tangle of new blood vessels feeding the tumor, despite the fact that the cells in the center are probably in a hypoxic environment. There's an effort by the system to produce fuel for this tumor to continue to grow. And lastly, we have tissue invasion and metastasis, which is actually quite a complex program. It's a series of steps that allow cells that are missed in this whole process to find themselves new homes in different parts of the body through the bloodstream, circulation, and distant sites where they can set up and colonize and produce subpopulations in places other than where the original tumor started. Those are the hallmarks, and those phenotypes are important. They're going to be really important for today's discussion. And I don't expect you to memorize them if this isn't like something you do every day, but I want you to at least have a sense of what those phenotypes represent. And we call them phenotypes because there are many different ways in which a genetically mutated cell could manifest one of these phenotypes. It's not a single genetic mutation that might contribute to genetic instability. We see in breast cancers one type of DNA repair defect that will result in genetic instability. We see in skin cancers, perhaps there's genetic skin cancers, a different type of defect that can result in genetic instability. So you can have different genotypes that power these phenotypes. But this framework does a nice job at saying, okay, we've got different genetic variants, but all of them have genetic instability. And we have different genetic variants, but all of them have this dysregulated metabolism, et cetera. So that's why these phenotypes are powerful, because they give you a way to think about all cancers have these things in common. When we talk, I'll get on a little bit later about chemicals, but what we're talking about is understanding that these layers of dysfunction are important. And cancer is not a monolithic thing. Chemical X makes cancer. Boom. It doesn't work like that. Cancer, we know, is a multi-stage, multi-step process that can unfold over time. And understanding these individual phenotypes is an important part of appreciating what toxicology is going to have to grapple with to deal with the disease and how it might unfold. So under toxicology and risk assessment, in the late 70s, we came out with the Ames test, or Bruce Ames developed the Ames test, and it became something very important. We discovered that a high percentage of carcinogens were mutagens, and they would damage a cell and create this initiation for genetic instability, and that became a nice way to screen for things. Well, if we could screen for mutagens, maybe we can keep carcinogens out of our day-to-day lives. Unfortunately, part of that legacy, because it's been for so long, is that we've thought of chemicals as things that cause cancer. And in the project, which I'll talk about later, we tried to ban the word carcinogen. I didn't want people talking about carcinogens. It's not that carcinogens aren't important. They are. We don't want single chemicals that could cause cancer in our lives. We would all agree on that. But what I'm going to talk about is, well, we might be interested in chemicals that can enable some of those phenotypes. We don't necessarily only care about chemicals that themselves can cause cancer. We might be concerned with multiple chemicals that might contribute to producing those cancers. So we have two things that you probably are familiar with. We have mode of action and adverse outcome pathways. For those who don't do toxicology every day, I'll just briefly touch on them. It's important you understand these two definitions. They're very close, and they will be really important for this afternoon's discussions. Mode of action is defined as a sequence of key events and processes starting with the interaction of an agent with a cell, proceeding through the operational and anatomical changes that results in an adverse outcome. For example, kidney cancer. So if we could say, if we had a chemical and we knew that exposure to that chemical would act on a cell, and we could follow the steps that went through what happened next, and at the end we had kidney cancer, we could say that chemical has that mode of action. That's a nice model, but it's really simple model, and there aren't very many chemicals that can do that. Adverse outcome pathway, and there's been a huge amount of momentum on adverse outcome pathway, suffers from the same limitation. It links a molecular initiating event, something that happens at the beginning, with an adverse outcome at the end. And the reason that I call those limitations, if I can just jump ahead here, is that, well, currently the mode of action is unknown for many chemicals. This is sort of something that's been under development for the last 10 years. The World Health Organization, with the help of industry, has been focused on developing mode of action rationale. It's become part of OECD directives for countries looking for chemicals to look for things that have common modes of action. If we had 2 chemicals or 3 chemicals that acted on the same pathway, through the same processes, the same adverse outcome, well, we better look at those cumulative effects, especially if we're establishing safe dose levels for the population. Makes sense. But in a lot of cases, since mode of action doesn't exist, if you look through EPA documentation, for example, and you look at registration documents on many chemicals, when it comes to cumulative effects, it says the mode of action of this chemical is unknown, and therefore, we don't know of any other chemicals we would group this with in terms of looking at cumulative effects. And that's true. It's just an unfortunate reality. That's where we are in the scheme of things at the moment. And so that's a bit of a problem. The population, of course, is exposed to a broad range of low doses of chemicals, some regulated, some not. Some carcinogens at levels that we think are safe, some that are non-carcinogens that have biological activity when you're exposed to them. But in general, the dose response thresholds on things that are regulated are all calculated based on when something causes cancer. So if we have a chemical and we know it causes cancer at this dose, let's set a big safety threshold, and we can now expose everybody to it at a much lower level with a safety threshold of 100. We can say that it's okay for everybody to be exposed to. Unfortunately, the dose response threshold, the point of departure, where we no longer, we say below that it's safe, there's no cancer at the end, doesn't mean we've reached the dose response threshold for the other things that chemical might do that might enable phenotypes, for example. It might contribute to some other aspect of carcinogenesis at a much lower dose level. So that becomes a bit of a problem because there's no recognition or a way to deal with the cumulative effects of all those low doses of chemicals acting maybe in very important ways on cells and surrounding tissues in ways that might be enabling of the carcinogenesis process. That's the sort of fundamental shortcoming that we're facing. If you look at cancer therapeutics, so it's kind of unfortunate. I'm very interested in environmental health, but most of the money, or large percentage of the money, goes to therapeutics, and they've been racing ahead and driving a lot of the fundamental basic science in cancer biology. And we've got chemicals in the therapeutic side of things that act on almost every single hallmark. So the way of approaching the disease, how do we shut down the disease, is to come up with therapeutics that can act very precisely, these very targeted therapeutics for personalized cancer medicine, on pathways that are deemed critical. So if we can shut down this pathway or that pathway. But as everyone knows, the big Achilles heel of most therapies is heterogeneity. And so in the clinic, people use multiple chemicals acting on different pathways to try and deal with multiple cell types that are enabled in different ways. But the point is that there are very few people who believe we're going to find that silver bullet. Nobody's looking for one chemical to shut down cancer. Actually, we do, but we've seen just from experience that that's not very successful or realistic given the nature of the disease. But my point is that often the way we shut down even with one chemical is by acting on one aspect of cancer. So the question that I would say, if you flip that over, say, why should we expect a single chemical will enable the disease, right? Why should one chemical cause cancer? Because now we're expecting one chemical that we know acts mechanistically. You know, the studies that lead to drugs involve docking mechanisms and receptors and pathways, and there's a lot of analysis as to what it takes to create the right molecule to act on a particular part of the cell to get the right kind of response. But if we're talking about environmental health, why should we think that we're going to have one chemical that's going to produce everything that's needed to enable that entire set of phenotypes that we know of now as the hallmarks of cancer? It seems unrealistic. And then from the work that's been done on low-dose endocrine disruption and chemical disruption at low doses, we've seen a lot of interesting effects at very low doses that are environmentally relevant. We know chemicals can have effects at low doses that are interesting and biologically important. They might not cause cancer, but we could have actions on cells. And the interesting and the most challenging part of cancer is that cancer can be initiated with one immortalized cell. You know, we have 1 trillion cells in our body or something like that, and one cell that goes rogue and becomes fully enabled can start creating copies of itself, and now you've got a problem, right, of epic proportion. So you can see that this question of living in a soup of chemicals that we're exposed to every day that might potentially be capable of enabling these various hallmarks in any given cell or tissue where they act in common and in concert is a question that is worth exploring. That was sort of the genesis of the project that we undertook. I was going to mention the bits about the environmental carcinogenesis being a little bit underserved over the years with the emphasis on lifestyle. Interesting, I think a lot of that came from some of the work in the early 80s, Dahl and Peto's seminal paper on cancer and lifestyle. And we were left with the impression that chemicals in the environment were a very small part of the problem, 2 to 6% or something like that. And a lot of emphasis was placed on what we could do from a lifestyle. Richard Dahl is now passed, but Richard Peto is still alive, and he wrote the foreword for this 2011 paper. And it was done in the UK to revisit that same question. What can we say about what does the epidemiology tell us about how much lifestyle factors have to do with cancers? And the answer was 43%. Now that's a stunning departure from where we were 30 years ago. They could only show with current data 43% of the problem was lifestyle related. I sent a personal missive to Richard Peto by email and said, what's the other 57%? You know, we know that genetics is probably a few percent. We've seen enough studies to know that there are people with genetic defects and as relatively small percent. But 57% is a big number, and his reply was fairly brief, but it was something like, it's probably other avoidable causes that have yet to be discovered. You know, what is that? That's a big, 57%. And I think this number doesn't get enough airtime. I know when you go and look at the epidemiology, NCI did a study on what does current epidemiology tell us, and in the United States we don't have the same quality of data, and there are problems with historical studies that haven't allowed them to draw conclusions. The paper that was done on it at the NCI said we can't draw any conclusions because the studies are flawed in a number of ways. So we're left at a point where this is definitely important. What do we do about it? I started this project on my own. Those were conclusions I drew 5 years ago. I sort of had a light bulb moment. It was like, wow, we're in a bit of a mess. We've got a environmental toxicology approach that's sort of out of sync with what we now know from cancer biology. And I talked to a number of cancer biologists about this problem, and most of them didn't quite understand what was going on in toxicology, like they're two different worlds. I approached Theo Coburn because she's in low-dose research, and she sent me to Michael Gilbertson. Where are you, Michael? Thank you. My wisdom in this project. Michael has worked for 20 years with Theo on low-dose endocrine disruption, not directly, but as a colleague or with common interests, and they work closely for a very long time. Michael has followed endocrine disruption to this day. He's still very active in what's going on, and he's been a huge asset and resource in this project. But what we did was we started Getting to Know Cancer, which was an NGO. We started it on nothing. We wrote it on nothing. It was a shoestring operation. Michael and I, both volunteers with no funding. We formed an advisory board, volunteers, scientists, 30 or 40 of them that all looked at this problem and said, yes, this is important. I recruited 2 top-tier cancer journals who said that they would fund special issues for 2 projects. I'll mention the other project just so you understand. I sent a spam to 40,000 cancer scientists, 1 email, and was reprimanded by my work for doing so. And we had 1,000 scientists respond to say, this is a great idea, I'd like to be involved. I picked team leaders, and I said, you can pick people that you work with, former colleagues, or anybody off this list of people who have put up their hand to say we're interested. We formed 24 teams for the 2 projects. And today we're going to talk about one of those 2 projects. I will just briefly mention the other half of the project before I move to this next slide. The other half of the project is a mirror image. Maybe I'll better explain this one first. Our goal was to use cutting-edge cancer biology to inform toxicology and risk assessment. That was really what we started to do. And really to summarize in a coherent way, in a high-impact mainstream cancer publication, what I just told you. We wanted to say that mixtures matter, and that we need to consider what their impact and effects might be. That was what we were trying to do. And we wanted to put it in the literature in a place where it would be undeniable. And so we produced a 170 author or something capstone paper with William Goodson as our lead author. And our goal was to draw attention to this issue. That's really what we set out to accomplish. I will briefly mention the other half of this project was a mirror image of this project. It was 12 teams focused on the hallmarks, finding non-toxic chemicals that would be useful from a therapeutic standpoint that had been written about in the literature that could be combined into benign mixtures that would be used to deal with the problem of relapse, because most cancers, relapse is driven by heterogeneity of cancers. And so we know that in the clinic, the limiting factor is toxicity, and combining chemicals therapies quickly becomes toxic. You can't combine more than 2 or 3 therapies and the patient is overwhelmed. So we wanted to be able to combine 70, 80 chemicals that all had no toxicity or low toxicity, using mainly chemicals that had been identified in plants and foods that were well tolerated by people. And we're producing a paper that will be out probably within a month, a capstone paper with another 170 authors who have all produced underlying papers as well in a special issue that's coming out in Seminars in Cancer Biology. That's the other half of the Halifax project. So in the Halifax project, we used teams, 12 teams. We had team leaders who were mainly cancer biologists. My thinking was that we needed to start with the biology. What does the science tell us so far? We tried to include environmental health people on every team. We wanted toxicologists, we wanted endocrine disruption people. Every team we tried to recruit people that could bring some of that perspective to the project. I limited their choice to 10 pathways because these domains are big. And I said, I just want you to pick some of the most important pathways in your area. Don't do more than 10. Because we're not solving the problem, we just need to explain the problem so other people understand it the way we understand it after having this discussion. And pick 10 chemicals of interest. They could be things that have been written about, things that have been, we were looking for chemicals that there was a good body of research on, that were ubiquitous in the environment, unavoidable, things that are in your blood now, that's probably in tissues that are in you already, that are selectively disruptive, not mutagens. We didn't want indiscriminate damage. We wanted chemicals that created specific types of adducts, or chemicals that acted on a particular receptor, or that could act in a particular manner to drive those phenotypes that we talked about earlier. And I didn't want to focus on things that were lifestyle related, because that's ground that's been trod over for the last 30 years, and not carcinogenic. Now, many people pointed out in the workshop, hey, you know, carcinogens are just as relevant. If we create a safe level for a carcinogen based on a point of departure, and yet we're interested in what kind of mechanistic effects it might have at low dose, they're just as dangerous in the environment. We should be looking at those. Those are obvious candidates, things that we thought were safe at the levels that we've exposed the population to. But I really wanted to create something that would capture people's attention. Not in an alarmist way, but I wanted to say we can talk about non-carcinogens and, to the point, subcarcinogenic carcinogens, subcarcinogenic exposures to carcinogens. And those effects at low doses are what we wanted to discuss. What are we really dealing with, and is this a problem? So we gave the team topics that are aligned with all the hallmarks and included genetic instability. I forgot to mention inflammation. I jumped over it in the first slide. But the inflammatory system, which involves the stress axis, the HPA axis, as it is also referred to, the cytokine network that produces the inflammatory cascade that results, and all the mediators that are involved in producing inflammation, are also known to contribute substantially to enabling these various phenotypes. And if you were to do surgery on cancer, you'd discover an inflammatory environment. There's a lot of inflammation in the tumor microenvironment, and that inflammation helps drive a number of these phenotypes forward. And we've now got good pathways and signaling, and we understand what aspects of inflammation act on what aspects of these various phenotypes that we know are enabled in cancers. So inflammation was an important area as well. And we added one other team, which we called cross-hallmark effects, anticipating that we might pick chemicals. I'll take any one chemical that we thought was interesting from a low-dose perspective, from the way it acts on molecular biology, anticipating we might pick that later as something we want to do testing on. We said, well, if one team picks it as something that is an instigator of this phenotype, and they think it's relevant at low doses, and there's data to support that, and we're interested in doing further work on it, it would be nice to know whether there's been other research done on that same chemical across all those other hallmark areas. So William Bisson, who's here, headed up a cross-functional team of 24 people, or 22 people, 2 in each area, who then looked at that chemical and said, is there anything in the literature that says that that chemical's been looked at from this perspective or from that perspective? So we have sort of a framework and a starting point for future research. We weren't saying that if the particular chemical acted on 7 of the 10 hallmarks, then it was a carcinogen. That wasn't our goal. We just wanted to be able to document what's in the literature now about those chemicals that we studied, so that people who were deciding to do work on them later could think about them, not just from the perspective that they had been chosen to be looked at, but also from the perspective of the other hallmarks that are relevant as well. So I won't go very deep into the findings, but the teams found 85 examples of chemical actions on key mechanisms that had been demonstrated at environmentally relevant low dose levels, the kinds of levels that we're exposed to. When we looked at dose response data, and we took those 85 examples and said, well, what do we know about dose response? Is there a threshold here that makes this safe? 22 of the 85 examples that the team picked turned out to be chemicals that had been tested that had a point of departure. There was a threshold, and at low doses, there wasn't any apparent action on the mechanism they thought that chemical was going to be disruptive of. But on 50 of the 85, which is a large number of chemicals, 50 chemicals out of the 85 that we studied, they found effects that had been shown in vitro, in vivo. The data's in our paper, but the point is these effects had been shown at dose levels that were relevant to human exposure. So we're exposed to them. They act on these mechanisms that are key important drivers involved in cancers, and they are believed generally to be not carcinogenic. So what do we make of the fact that we have the collective exposure experience that we have, given what we found? There's 22 of the 85 for which no dose response information existed. We don't know if they're active at low doses. We do know that they can make that action, but it may have only been demonstrated at one dose. There's no pattern of dose response to allow us to make that assumption. So low dose effects on key mechanisms and pathways. You can't probably see this slide, many of you. I included it because you'll probably get access to my slides so you can look at it. This data is in the paper in a different form. It's tabularized, but just a quick snapshot, you'll see that there are a number of chemicals in each of these areas that have some activity on all of these phenotypes. Now, I will emphasize that doesn't mean that all those chemicals are acting on the same cell in your body, right? We don't know that. But we also don't know that they don't. It's a question. You know, we've got chemicals that are capable of these effects at low doses that are environmentally relevant. We're all exposed to them, and they're capable of acting on these very important driver mechanisms in these key pathways that enable these phenotypes within the hallmarks framework. The hypothesis we emerge with is one that says that low-dose exposures, the mixtures of chemicals in the environment, may be acting in concert with one another to cause cancers. Well, people have been saying that for years, haven't they? I mean, that's what some might say. But this is the first time that anybody has ever gathered this information together and displayed it in one spot. There have been bench scientists doing study after study after study saying, hey, look, I found this chemical that we're all exposed to that acts on this mechanism, and the study's over and it goes nowhere. It just sits there. And we wanted to say, well, what do we really have now? Out of all the research that's been done from an environmental standpoint, what do we know? And really, this is the first time we've been able to show what we know. The teams, many of them had many more chemicals in their list. They, you know, some of the teams stacked up 30, 40 chemicals, and I was like, no, just keep it simple. This is not about pinpointing single chemicals or creating any kind of definitive list. Nicole Kleinstroyer brought to the table the ToxCast data, which is a lot of chemicals that are in our food supply. That was enormously helpful, but it was only the phase 1 data. There's more data coming behind that. And we're getting to the point where there's lots of information out there. The problem's been well described in the paper by Bill Goodson, myself, and our colleagues. And our goal was really to bring it to public, not the public, but the scientific community to say, hey, look, this is an issue that we need to be thinking about and thinking about ways forward. I'm going to jump back. How am I doing for time? Somebody help me. Okay. I only have maybe 5 minutes or so. I want to jump back to what OECD provides for guidance to nations around the world now under the World Health Organization International Program on Chemical Safety's guidance, and it's all based on mode of action, and that is that the risk from cumulative effects focused on groupings of chemicals that produce a common adverse outcome. So only things that cause kidney cancer will only be looked at for cumulative effects with other things that cause kidney cancer. That they have to act via common sequence from that initial initiating event all the way through to cancer. They have to be the same before we worry about whether or not we're exposed to them both simultaneously and what safe dose levels might be. And they have to act on the same tissue. But, you know, the phenotypes that we see in cancers are much more nuanced and complex than that. So this is a very crude model. I don't want to say that adverse outcome pathway or mode of action is irrelevant. It's good for single agents that are complete carcinogens. What we're talking about is that there are many chemicals that can have a contribution to play at the kind of dose levels that we're exposed to, and we need to have some way to deal with mixture effects. It's much more complicated than what adverse outcome pathway would have us believe. And there's an incredible amount of momentum in the toxicology community on adverse outcome pathway. And to Linda's point at the opening here, I hope that people can see just after this short discussion, that's really last decade's way of thinking. It's an outmoded, in terms of its ability to deal with this complexity. It's good for what it does. For single chemicals that cause cancer in the same manner on the same tissue, fine. But when you start talking about the many ways in which you could envision mixtures having synergistic effects, take a lesson from what the people in therapy are doing. They don't expect to be able to act on the same mechanism with one chemical or a range of mechanisms with one chemical. They expect to be able to act on single mechanisms with single chemicals, and when they need to get to another mechanism, they pick a different chemical. And if we think about it from an environmental health standpoint, that we should be thinking about how we might be enabling those phenotypes with different types of chemical exposures. If you'll indulge me for a minute, I want to tell you a short little story here. In 1987, at the age of 24, fresh out of university, I found myself in the Air Force taking pilot training, and this little plane was what I flew. As a single 24-year-old, life couldn't get any better. I want to point out 3 safety systems on this jet that are interesting, and you'll know why in a minute. The reason is that these are safety systems that are really critical. They don't get used very much. If you're need to slow the jet down quickly, which doesn't happen very often, and I mean like an emergency of some kind, there's a speed brake, these big flaps that go straight out into the wind stream to slow the jet down. Basically, we ignored them in training. Nobody used them. I mean, the people doing our snowbirds, which are like Blue Angels demonstration team, they use them because when they're forming together, they would slow their planes down to get together, use them quite frequently. We just ignored the switch completely. Giant tail rudder on the back of this plane was specifically designed on this model so that if you ended up in a spin, if you get a high altitude and the nose gets high and the plane stalls and starts to fall, it falls like a leaf in circles. It does this wild spin, and you have to have a way to counter that spin, and so it has a big rudder on the back, and if you stick your foot hard on the rudder, you can stop it from spinning. But even though when you learn to fly planes in like a little Cessna, the rudder's really important because as you're going downwind, you have to be traveling on an angle, and the rudder is really critical. In a jet that goes 400 or 500 miles an hour, generally you ignore it. When we went into training, they said forget the rudder, except when we're doing spins. Use them as footrests. You know, the 2 foot pedals, just put your feet on them, forget about them. You can ignore them. And then we had ejection seats. We were trained in detail on every aspect of the ejection seat. The canopy blows off in an emergency. If you pull the handles, the canopy blows off. The seat fires out with a giant explosive. There's a drogue that deploys the seat away from the pilot, and then the pilot chute deploys, and they call it seat man separation. And you become safe. But frankly, most days you just it was like you didn't even know it was there. I mean, there's no reason to use it unless it was an emergency. So if any of you know me, I'm a bit of a unpredictable at times when I was 24. One day, I was flying the plane in early solos, and I decided I was going to do a maneuver that I hadn't been trained on. It's like a few chapters ahead in the book, literally. There was like a few chapters ahead, but I heard people in the mess hall telling me about, this maneuver. It was a half loop. It was a pull through. You go up to high altitude, you roll the plane inverted, and you pull on the stick, and you do the backside of a loop, and you come out at the bottom. It's like, woo! So I had no training in this, but the jet was pretty capable. You could do anything in the jet. It was fully aerobatic. And so I went up there, and I went up to like almost 40,000 feet, and I'm doing 400 or 500 miles an hour. I got it on full throttle, and I was like, this is really fun. And I roll the jet upside down, and I think, okay, now I'm upside down. I pull on the stick, and I started headed towards the ground, and I was headed straight at the ground. And I was pulling on the stick as hard as I could, and I could not get the nose to come up. I was like just in a straight dive. And, I should have read a couple chapters ahead in the book. What I would have learned is that when you start at 40,000 feet and you go downhill, you pick up speed. And it didn't dawn on me. The speed was going higher and higher. I was at full throttle when I started. You're supposed to start this maneuver at a slow speed so that as you go down the backside of this loop, you pick up speed. Anyway, there's all this wind buffeting on the cabin, and I was not panicked, but I was thinking, what the hell do I do here? Because I don't know what I do. So I pulled the throttle all the way back, and I remembered somewhere in the back of my mind was this little switch on this handle that you could pull to pull out the speed brakes. And when the speed brakes come out, the nose came up, and it was like the plane slowed down and everything was normal. And I was like, oh my God. Okay, why am I telling you this? Because this is a system that's designed to keep the pilot alive. It's to avoid death, really. There's got a lot of systems to keep the pilot alive. And there are a lot of systems that don't get used all the time. And I would argue that the cell has all these layers of safety systems that keep the cell from becoming cancerous that we rely on every single day. I'm going to show you just how these phenotypes help us think about why these systems are so critical. They are systems that often don't get used, but they are absolutely essential to making sure you don't get cancer. We know that if you eat French fries, it's in the news, you get mutagens in French fries that can act in a way that might be similar to what a carcinogen would produce, but we don't all get cancer from French fries. There might be some percentage of people, but the mutagens are often dealt with initially with DNA repair. Job number one, you go out in the sun gives you damage, DNA repair fixes it. People that have XP, xeroderma pigmentosum, pardon me if I got that wrong, they have a genetic defect in DNA repair. They have to spend their life indoors. They're deprived of sunshine because if they go out into the sun, they get cancer. But we have chemicals that can disrupt DNA repair. EDTA is in everything. It's in foods that we eat. It's in all kinds of products. It's in sunscreen. It's in after sun. It's a DNA repair disruptor. Now, is it effective at low doses? How does it work in mixtures? I don't know. I'm just saying I don't want my DNA repair to be broken. I don't want things in my body that disrupt DNA repair. It's a system like in the jet that doesn't get used, or that if it does get used, I'm not even aware it's happening. It's doing its job and I don't see it. If it wasn't there, I'd be worried, just like the people that have that genetic defect. We have tumor suppressors, P53, but we have chemicals that act right at P53, create adducts that make it dysfunctional. P53 is a tumor suppressor that's disrupted in 50% of all cancers. It's been on everybody's radar for 3 decades. And yet, we've got chemicals that can disrupt P53, and as long as they're not carcinogens, nobody cares. And I don't mean nobody cares, I mean if they pass the test of carcinogenicity, they're not carcinogenic, or if we can find a level at which they are, and they're safe at a lower level not to cause cancer on their own, then we can expose everybody to it. But I don't want things that I'm being exposed to be disrupting P53, for example. Programmed cell death, senescence, the ability of a cell to shut itself down if it's made too many copies. I don't want chemicals that can disrupt that from happening. If I've got chemicals that are making too many cells that are making too many copies of themselves, I want them stopped, right? The epithelial mesenchymal transition. There are examples of phthalates, which we're all exposed to, or multiple chemicals in the phthalate family that can enable this transition in tissue invasion and metastasis, where cells transform from their normal differentiated cells into this sort of naive, undifferentiated, immortalized, motile phenotype that can move to different parts of the body and set up new sites. There's been a lot of groundbreaking work done in the last decade on EMT, epithelial mesenchymal, and there are chemicals that we know that can act on that and produce that transition, which is actually a very complex set of things that have to occur, but one chemical or multiple chemicals are capable of causing it. So do we need to worry? If I was a cancer patient and I already had cells that had formed a tumor, shouldn't I be worried about exposures that might help move that process along by enabling metastasis, which we know is usually the endpoint for most cancer patients. Those are things that are inside the cell. Outside the cell, we have completely remote systems. The immune system, which is distributed, the thymus, and different places in your body where immune cells are produced and created, and the tissue interactions themselves. We've been using 4-ball ester, TPA, for 2-step initiation promotion experiments for a few decades. It acts on the gap junctions between one cell and its neighbor. And if you disrupt the signaling between one cell and its neighbor so that it doesn't think that it has a neighbor anymore, it starts to make copies of itself because it thinks there's something missing. So if we chemically can act on this, we can produce proliferating cells with one chemical. And we realized quite quickly that gap junction intracellular communication disruptors, which is what 4-ball ester is, are dangerous. And we've noted that. But that's only one example of a mechanism where people got, whoa, we need to be careful not to be exposing ourselves to that because of what occurs. And then we have inflammation, which is the stress axis and all of the big cytokine network and the inflammation constituents that cause inflammation, that are also outside the cell that we need to worry about. Is our body capable? We have adrenal disruptors, lots of them. The EPA has got all kinds of data of things that disrupt the adrenal cortex. We do radio tracer labeling studies of chemicals that end up in the body, and we've got examples of chemicals that are disrupting the adrenal cortex, which is where cortisol is produced, which produces cortisone, which is what shuts down inflammation when it needs to be shut down in the body. And we don't really pay attention to whether or not the adrenal gland disruption has anything to do with cancer. It's not even on anybody's radar, and it's in a completely separate location from where the cancer might originate. So when we talk about only being concerned with cancers that cause chemicals that cause the same type of cancer and the same type of pathway progression from a single initiating site and one mechanism for single chemicals, and that we're only going to think in adverse outcome pathways or mode of action thinking about looking at cumulative effects when we have all those things in common, and we'll ignore all other mixture possibilities. The argument that we've made in the Halifax project is that we're really missing the boat. That's kind of what I'm saying here, so I'll just jump past that. I should avoid this too. This is really saying, this is my equivalent of we only care if the plane crashes. That's under adverse outcome pathway. I would say if I went out to sign out the plane and the ejection seat didn't work and the speed brakes were busted and various things were missing, I'd be thinking, I'm not sure I want to fly that plane, because when I get out into the real world, other things might happen, foolishness perhaps, and I may need some of those systems to come to my rescue. And that's the way I think about exposures. Our cells need those systems. They're critical for them to avoid the immortalization process that we know happens. The Halifax project is all about saying that the cumulative effects of mixtures matter, that cancer biology tells us we have to be concerned about low-dose exposures that can enable hallmark phenotypes. It doesn't matter if they're the same sequence or dissimilar sequences, if they're active and they're acting on the same tissues, or even different tissues. In the case of inflammation, in the case of the immune system, we need to be thinking about effects system-wide, not just at the point of initiation. And a range of different chemical types. There's a practice at the EPA of saying, well, let's look at the organophosphates as a group, or let's look at the triazines as a group. And they're thinking of chemical structure similarity as meaning that might mean something. I would say if you started from a biology standpoint, we'd be less concerned about chemical structures and more about chemical actions on those structures. And there's been people who've written on that. I mean, that's not new. The next steps, what we really hope to accomplish in this project was to be able to use low-dose chemistry to help unravel carcinogenesis. I haven't said much about it here, but all the teams struggled with the fact that all the research literature is on cancer. What happens in that latency period? Why does it take 20 years? What's the process? Forget the theories of somatic mutation theory or tissue organization theory, but we can use chemicals. We think that we can use chemicals to be much more nuanced at this than we ever were with initiation and promotion. People in the 30s, there was a scientist in Buenos Aires who was doing, Raffo was his name, who was doing two chemical, like he would create damage and then he would put a chemical and he could get cancer on rabbits' ears. We're way past that. Our biology has raced us ahead. Now we should be thinking, how could I enable phenotypes in stepwise progression? What if I started this phenotype in cells first with this chemical and then got this phenotype underway, and then a next step, and what does it take to get this process? It's self-promulgating once it's going. We know that from people that get it from genetics. But we should be thinking about how we could use chemicals that we have at our disposal, what we've learned about in low-dose research, to be able to produce carcinogenesis in some kind of sequences that allow us to better understand how the disease might unfold. That's something that's not understood well. Our team struggled with it. When they went to the literature, everybody came back and said, where's the stuff on what's the process of the unfolding of cancer, which is really critical if we want to talk about carcinogenesis. We don't understand that process very well. There's some basic science that's missing there that has to be done. We're hoping that the Goodson paper will be helpful for the next steps. We managed to get 178 scientists, 12 teams, all through peer review, and the paper all out on time. Maybe it wasn't quite on time, but we did get it done. I was going to say we didn't lose anybody along the way. There were a few people lost along the way, but we really did finish it, and I'm hoping that it will be the right platform for people to take this the next step. Thank you. Am I like way over time? You want to move questions to the end or something? Sure.
Pardon? Hi. I don't know. I'm Bill Goodson. I trained as a surgeon. I did wound healing research at a time when people were trying to figure out what PDGF was. Worked with wound angiogenesis and tissue perfusion with Tom Hunt. And about the middle of the 80s, I found myself with my clinical practice entirely breast disease, and my laboratory work involved in wound healing, and I was asked to be involved in a cancer research project, and it was kind of like the two icebergs were going like that, and I figured I was going to end up in the water, so I jumped over to breast cancer, which is where I've been since. I was trying to think of what my role is in this discussion this morning, and I'm still a little vague on that, but what I think is most important is to realize that as toxicologists and people who are specialists in the cancer biology, you have one perspective, and as a person who talks to oncologists, I have a different perspective. I deal with this all the time. I've had conversations, I won't give names, but I've had conversations with the president of ASCO, the American Society of Clinical Oncology, the pinnacle of medical oncology, and the conversation kind of runs, Bill, this is very interesting. There may be something to it. You'll never be able to prove it. Let's move on. And that's very disappointing, but it really gives me partially the reason I'm going to talk about what I'm talking about today, because I'm going to talk about sort of the things that are not in the paper, but which I think are very important as we think forward, because part of this is going to be a question of convincing ourselves. Part of this is going to be a question of convincing regulators. And most importantly, it's going to be a question of convincing the clinicians who deal with cancer all the time. Because if we don't have their buy-in, we've got a problem, and we won't really get very far with it if we have large organizations who are saying that's all very interesting, but so what? So the end of the first day of Halifax, we had several hours before we got together and had an evening program, and I was down by the bay at Halifax Bay, and there was a cruise ship leaving. And it's absolutely fabulously clear water. It was a glorious day, and if I could go back and get that day again, I would. It was a beautiful day. And I thought about it, that this ship is heading out into one of the most perilous pieces of water in the world, the North Atlantic. When the North Atlantic gets angry, it's a real problem. And there are many things that come together in the North Atlantic that can come together, as we may have all seen the movie or heard the story of the perfect storm. And it may not be one thing, it may be another thing, but everything can combine the wrong way at the wrong time, and then you've got a problem. So picking up from where Leroy left off, is that we aren't exposed to one chemical on a daily basis as a chemical A on Monday and our Tuesday chemical and our Wednesday chemical, etc. And that's an extremely important concept because we're exposed to varieties of chemicals and we may not be exposed to the same mixture of chemicals every day. We can have days when we don't see A and days when it's all B and C and D and stuff like that. So we're in this gamish all the time. That's the first thing that I take out and pick up point. The second thing is that there really are hallmarks of cancer. There are all of these characteristics that exist, and I put up P10 here because P10 is sort of the brakes on this. It is those little air brakes that flip out the side of the jet plane, and it's important too. It's not really a hallmark, but it's very much involved in controlling sustained proliferation and evading growth suppression. Now, the reason this becomes important, or where you go with this, is that there are chemicals that can affect these. Now, this happens to be bisphenol A. Bisphenol A can cause sustained proliferation as long as it's there. It can cause evasion of growth suppression, i.e., it can make cells not have apoptosis when they should and resist cell death. But it also, I misspoke, I got out of order here. It also can suppress P10. So cells which are exposed to bisphenol A will lose P10. And in at least one of 200 samples that we've looked at, we have found replicative immortality in cells which, after they were exposed to BPA, they continue to grow like gangbusters. 1 out of 200, when you think about it, and I'll come back to the statistics of what we're talking about, that actually is probably a fairly significant number. On the other hand, for at least 3/4 of a century, plastics, chemicals, and everything that man can make has been very much a part of our lives. And I don't think there's a way to turn back the clock. I'm not trying to turn back the clock. I do want to know what's going on in the clock. It's the same reason I'm sure that I will eventually be eating GMO food, but I'd like to know it. And it's the same kind of thing here. So I'm going to talk about two things. I'm going to talk about things that I think are very established, and I'm going to talk about things that I see as real challenges. And this is looking at it kind of from a clinical standpoint. We live in combined chemicals. Mixtures can be biologically significant. There are anthropogenic chemicals. Single hallmarks can predispose to cancer. You don't need to have all the hallmarks kicked off to be in trouble. The cancer rate varies by the risk factors, and gene expression is as important as gene mutation. And most of what I say, I'm going to assume that at least half to 3/4 of you know everything I say, so I'm not going to spend a lot of time explaining it in the kind of detail I would to first-year medical students. I'm going to talk about it and then talk about what I see as the highlights or the important points to take from it. So the first thing is we live in combined chemicals, and it would be carrying Cole and Newcastle to try to summarize, or for me to go through the NIEHS data, Statistics Canada data, the innumerable studies that have been done looking at all sorts of tissues and all sorts of blood samples. We live in that all the time. I always refer to what I think of as the 2-ton salmon study. A, I like alliterations. And B, I think this is an extremely important study because what you're looking at in the red lines are the content in farm salmon, and in the green lines, the content in wild salmon. And somewhere along here, total DDT, this is a log difference. There's a whole lot less DDT in wild salmon than there is in farm salmon, but I don't think there's a natural source of DDT. So there's a lot of DDT, and the farm salmon is mostly grown in areas around North America and Europe, some in South America, and oddly enough, South America, there's less DDT in the farm salmon, but most of it is in areas or downstream, so to speak, from the land where DDT hasn't been put on the land since God knows when. And we're still getting DDT in the salmon. So as I like to point out, what goes around hangs around. So this is a major issue, but this is kind of an established thing. The second thing is, as Leroy alluded to, mixtures can be biologically significant. The treatment of cancer with drugs depends upon mixtures. Now, most chemotherapy that started out, CMF, AC, and things like that, were drugs, anthracyclines, methotrexate, fluorouracil, ticytoxan. They're all drugs that interfere with the DNA. But what really changed breast cancer chemotherapy was adding a taxane, an antitubule agent, an anti-division agent. So all of a sudden you went from damaging the DNA of the cancer cell to actually interfering with the division of the cancer cell, which is a different process. So mixtures are very important. And if you look at the really exciting stuff now, It's the mixtures of drugs that are being used for metastatic cancer that are just sort of blow your mind away. The mixture of anti-metabolism and anti-signaling, and everolimus basically turns off the metabolic choke point for cancer cells, and the aromatase inhibitor cuts off E2 growth signaling, so you lose the signal at the beginning. So you're losing the stimulation to growth, but you're also cutting off the metabolism. And then the one that's really most exciting is this palbociclib, which is a combination of anti-proliferation and anti-signaling. So you decrease the estrogen growth signaling by downregulating the estrogen receptor, and then you come along at the very end and you kick the cyclin 4/6 in the teeth, and all of a sudden you have a very marked difference in what happens with the cancer. So this is the data with everolimus. It's statistically improved disease-free survival over letrozole alone. That's statistically significant, but it's not changing lives a lot. This is what's happening with palbociclib. And you're starting to see people with long-term where you don't reach the 50% recurrence rate on the combination of exemestane, fulvestrant, which downregulates the estrogen receptor, and palbociclib, which basically turns off cell division. So when you look at this, what you're looking at is the basis of chemotherapy. The basis of cancer therapy is working with a mixture. Now, if you fight cancer with a mixture, how in the world can you not turn around and say, well, if you can stop it, why can't you start it? And I think that's a really important point to bear in there. The next point is that there are chemicals that are clearly carcinogenic. And the important message here is DDT, powerful insecticide, harmless to humans. And this is a picture published by the National Geographic several years ago. It was taken on a beach in New York out on Long Island sometime in the late 40s. And as we know, not only does DDT accumulate, but DDT will cause breast cancer, or will cause an increase in breast cancer, and it seems to be related to the age at which people are expected. So this is the data which shows by Cohn working with this amazingly fortuitous database that was developed by Kaiser in the late 50s, early 60s, where it was going to be part of a study of fetal development. They drew all these blood samples, somebody stuck them in the freezer for 30 years, and they went back and analyzed the DDT levels. And the important thing they found was that in people who had reached maturity by 1945, which was when DDT came on the civilian exposure, it didn't make much difference. You'd always wonder why it looks like it goes down a little bit. But in the people who had not matured by 1945, less than age 14 in 1945, the highest quartile of DDT had 5-fold increase. Now, this is something that's looking at the parent. About a month and a half ago, this came out, which is the same data set, but what they've now done is they looked at the offspring from these same pregnancies. And yes, family history of breast cancer is a high risk ratio. Overweight in pregnancy is a little bit of a modifying effect the other way. But looking at the children 40, 50 years later, looking at the DDT level in the mother, it predicts breast cancer so that the highest quartile has 3.5 times the risk of breast cancer. So clearly, chemicals are in effect. But the other chemical, and it's the one that I think is greatly not understood in terms of what it is and for what the real lesson of it is the work of Edward Charles Dodds at the Courtauld Institute Middlesex Hospital in London. Now, Edward Charles Dodd looks like you'd expect an English professor to look like, all apologies to the English, and he set up the first clinical laboratory in a hospital in the United Kingdom. And in the late 20s, the King of England was sick, and he did a blood analysis and discovered that the King of England had low calcium. Now, we never figured out why, but it gave the king calcium. He woke up and he was fine. So this is one of those things that I don't know the rest of the story, other than there's a whole series of newspaper articles about calcium for the king. Now, Edward Charles Dodds was a person he was an endocrinologist and interested in that, and he took the observation that no less than 5 distinct substances found in nature. They'd gone through and figured out there were 5 different things that worked as estrogens, including estradiol and stuff like that. And this is him writing like 10 years later about it. And he and some of his friends reasoned, well, if one chemical can do it, and then you have a series of chemicals, are there other chemicals? So working with Wilfred Lawson, who was a synthetic chemist, they set about to test a whole variety of things. And that you can go look at these lab sheets. Phenanthrene, the basic structure for estrogen, negative results 5, positive 0. but dimethylphenanthrene is 5 positive results and no negative results. And you can go through this and realize he did this massive study, and what he was looking at was just whatever chemical they can dream up. I didn't put in some of the really elaborate structures that they even tried diesel fuel. But the thing that he found, the simplest structure that he found that worked as an estrogen or had an estrogenic effect was 4,4-dihydrodiphenyl. Now, this is a pretty simple chemical. In a follow-up to that, he actually was considering this as the carbon, and he was hooking various things onto various sides here to look at what their effects would be. And what he was doing the so-called, I'm going to get atoms and alums mixed up here. It's where you have a castrated mouse, and you give it the chemical, and then you do a vaginal scraping and look for keratinization of the vaginal epithelium as a marker of estrogenic effect. And so he published his papers on all this sort of stuff. But what he then did, which I think is, you think about this guy's mind, they had figured out the structure of estradiol. He looked at bisphenol A, and he publishes this paper where he says, well, we came up with this chemical, and he called it stilbestrol. And this is diethylstilbestrol. It is not estrogen, but it is the strongest estrogenic effect of any known chemical. And he dreamed that up before crystallography, 3D structures, or anything like that. He looked at this, and said, we can modify this, and maybe it'll work better. I think that was brilliant. But what this led to was this brief history of DES, which I think is very important. First of all, DES and Dodds get castigated for what happened with women and stuff like that. That had nothing to do with what he was working on. He was looking for a cheap, easy way to treat metastatic prostate cancer. And actually, from the 1940s until the 1990s, when the LHRH agonist came out, it was the go-to drug for metastatic prostate cancer. And although Smith Brothers, advocated using DES to quote, prevent repeat miscarriage, and it was proven useless in the 60s, and we had clear cell vaginal carcinoma in the daughters in the 70s, that had nothing to do with what Dodds was intending, although he got castigated. The BBC did a program about how horrible this drug was, and the proof that it was not at all on his mind is when the New Year's list came out for the knighthoods in 1961, when he became Sir Edward Charles Dodds. There's a little newspaper clipping where this guy's writing about who in the heck is this guy Dodds that never heard of, and oh yes, he's working with this gland that we don't talk about, the prostate gland. So that wasn't Dodds' idea. The interesting thing when you think about it is the irony of Dodds doing a study looking at the effect of DES on the vaginal epithelium of a mouse, and here we are 30 years later realizing that that is exactly what it does. The drug is doing what it's supposed to do, but it also is leading to vaginal clear cell carcinoma. They actually did studies. They looked at vaginal biopsies from teenage girls whose mothers had taken DES and found that about 70% of them have proliferative vaginal epithelium as teenagers. It's the fortunate ones who just have that and then others. But you could see this in the laboratory screening test 30 years before it was recognized as a clinical problem. In the 90s, we knew that there was more breast cancer in the mothers who took DES, and then several years ago, Hoover in the New England Journal showed that there are definitely almost 2 times as many breast cancers in the offspring. So something has happened. Now think about this as usually a 3 to 4 month exposure. The mothers get more breast cancer 50 years later. The daughters get more breast cancer 50 years later. It's kind of a staggering thing. Actually, the mother's about 30 years. The next thing, sort of a given, is that you don't need to change all the hallmarks, as far as we know, to have something that predisposes to cancer. A lot of single changes do predispose to cancer. BRCA1 and 2 is a double-strand break repair defect. PALB2 is positioner and localizer of BRCA2, or partner and localizer, excuse me, and those things are basically affect double-strand break repair. Lynch syndrome is mismatch repair, microsatellite instability. All of these are associated with fairly high risk of cancer, single gene mutations. The key thing to look at, though, is that Li-Fraumeni has nothing to do with DNA. Li-Fraumeni is a p53 defect. Check 2 is a defect in the management of P53, and both of them have an adverse effect on S phase and apoptosis. Cowden syndrome is the loss of the brakes that we talked about a little while ago. And the question that you got to think about is how in the world do they accrue the other hallmarks of cancer? Because if somebody's a BRCA mutation, they do have sustained proliferation. They do have abnormal growth. It isn't just that they're sitting there not repairing their DNA. They have picked up other stuff. And Leroy, I think, was the first person I ever heard say this, although I think other people think it too. It may look like chance until we know better. So with all respect to Kurt Vogelstein, I think that the issue of chance is something that we need to sort of wonder back, because we used to think that people incurred the wrath of God to become sick, and we've learned a lot since then. And the other question that comes up is why do some cancers occur earlier with the same mutation? Before we go there, I want to point out that the cancer rate varies by the risk factors. With the BRCA, it's 80%. The PALB2, which is a modifier of this, an enabler of it, it drops to about 35%. Cowden's is about 80%. Lynch is about 80%. Leaf-Romini is about 90%. But Check 2, which modifies Leaf-Romini, which modifies the P53, is only about half. So this is about half, and this is about half. That's puzzling. But you look at the other things that we're talking about, the diethylstilbestrol exposed versus not is just less than a twofold risk. DDT is about 3.7 in the offspring. But when we sit there and say, okay, that isn't all that much, the key thing to realize is, nobody's going to go out and sit down win from one of the Nevada testing sites. And the relative risk for the people of the Hibakushu, who are the survivors of Nagasaki and Hiroshima, is about threefold. They didn't all get cancer. There were a lot of people who did, significantly more people who did, but not all of them got cancer. And even when we talk about CT scans of the chest, the estimate is that only about 1 in 2,000 people who gets a CT scan of the chest as a kid will actually get a fatal cancer later in life because of the CT scan. It's not everybody, but we're talking about things that have very pronounced effects when they do have them. Now, the one question that always has to be looked at in this is the question from epidemiology, and that is that why are people with the same gene mutation getting cancer earlier in life? This is Mary Claire King. She talked about looking at the BRCA mutation carriers' risk of breast cancer, and people born before 1940 developed it later in life. People born after 1940 developed it earlier in life. And people have criticized this, but then Litton et al. And MD Anderson came up with the same thing. They looked at the second generation versus the third generation according to the birth year of the second generation, 30 to 39, that was basically the same, 52, age 55. But when you come down here to the second generation born in 70 and 80, you have the second generation develops cancer at an average of age 31 as opposed to an average of age 44. We are moving cancer forward in life with the same mutation. Something is going on. And finally, this is just last fall, looking at the PALB2, and it was again broken down, take the relative risk of 1 for people who had birth year before 1940, and then look at the people who were born the same mutation, born after 1960. You have 6 times greater risk of cancer early in life. This is breast cancer. It's a variation on the BRCA mutation. Now, the other thing that's a given is that about 15 years ago, almost 15 years ago, we had Science and Nature came out, and the world was going to change because in one of them, depending on whose genome you looked at, you had 26,000 or 31,000 genes. It was going to be a simple mathematical problem. This is kind of like, remember the statement about an artificial heart is a simple engineering problem. This is going to be a simple mathematical problem to sort it out. But in one of the accompanying editorials in Nature, Judson pointed out the critical point never to omit is that genes act in concert with one another and collectively with the environment. And it was known at that time, what has subsequently been figured out is that the epigenetics, everybody has always talked about chemical mixtures and everything else that affects the normal cell and all this sort of stuff. But the thing that's absolutely fascinating is that we start out life, basically that little egg and sperm are scrubbed of their epigenetic changes. And we spend our whole life accumulating epigenetic changes that turn off the genes that we don't want on. And then you come along, and this is the one paper Ambar, this is Langdi in one of the papers in the special issue, arsenic, cadmium, quinones, benomyl, persistent organic pollutants, and nanoparticles to start out the list are things which can interfere with DNA methylation. So the important point in this is that the things that are already established, we live in combinations, mixtures can be biologically significant, there are anthropogenic chemicals, the question is not whether, it's just how long the list is. There are single hallmark changes, cancer rate varies by risk factors, and gene expression is as important as gene mutations. I'd move on to now what I see as the challenges. First of all, we need better epidemiology. Secondly, we need better understanding of physiology. Thirdly, we need to look beyond xenoestrogens. We need better models. And we need to shift from similar structures to looking at the interaction of dissimilar structures. And we need to understand chemicals and epigenetics, because I'm convinced that the epigenetics is an extremely important part of this. It was alluded to a little while ago that we're told that there are a whole series of a whole series of factors that are argued to increase cancer. And when you talk to clinicians, they tell me, well, this is obesity, it's alcohol consumption, it's sedentary lifestyle. This is why we've had this increase in breast cancer from the 70s up through the turn of the century. And then there was a little blip down after the Women's Health Initiative, and they said, okay, we're going to take all these women off hormone replacement therapy. Anderson published this article when the curve had gotten here. You can go to the SEER data, take the data out, and put it in Excel. And the humorous thing to me is when you put it in Excel, you get a chart that looks exactly like Anderson's chart, so you know that Anderson did his chart in Excel, even to the coloring. But this number is now stable through 2009. I checked 2009, the SEER data is always several years behind. This breast cancer incidence rate for invasive breast cancer, skip DCIS, that's a whole other argument. Invasive breast cancer has settled in at a level that's about 25% higher than what it was 20 years ago. And we're told that, some of this is over-screening, the whole series of things. Women have changed, they're sitting around, they've taken hormones, they've taken birth control pills, blah, blah, blah. Well, I'm going to talk about that a little bit. But the place to start talking about that is to go back and look at Dahl and Peto's paper from 1981, in which there was also about a 30% of increase in breast cancer over the previous 20 years. And in fact, Dahl and Peto says that we have the increase in breast cancer because of more screening, where he literally coins the phrase of talking about biologically insignificant cancers that just happen to look under a microscope like other cancer. So he's arguing that we're misdiagnosing, we're overdiagnosing, it's sedentary lifestyle, it's alcohol, and it's weight. And the problem is that gives you this increase. But if you want to really get a perspective on it, shrink the scales, I love what you can do with a computer these days, make it so that your timeline matches and your y-axis matches. And what you see is that there is about a doubling in the incidence of invasive breast cancer. Now, I recognize that there are statistical problems in trying to merge these two curves, but I also think that until we get epidemiology that really does a better job with it, this is what's available right now. And I find it hard to believe that you've gone from 65 per 100,000 to 128 per 100,000. And this actually, the 2008 number ticks up to 129 per 100,000. This is not a downward curve. It's very hard to believe that you have a doubling of breast cancer strictly because you've overcalled it, and all because of lifestyle. So I think that there are some real issues there, but if you really want to go into it, this is an article from Holford at Yale, which I think is a great article. He said, okay, if it's all gone up because we're now recognizing things that weren't there, he said, fine, let's look and see what was there before. So he's gone back and looked at autopsy series from 1933, 30 to 39, 35 to 44, and large numbers. This is his calculation for the false negative rate. But what you have is that in Los Angeles County, 33 to 37, 8,080 autopsies, there were 66 breast cancers found. If we're going to double the breast cancer incidence on the basis of finding previously unrecognized cancer, there's got to be a reservoir from which you can draw it. And there's no reservoir, there's no evidence of a reservoir from which to draw undiagnosed breast cancers. And finally, the most important part of this is that when you look at the little line that Anderson did with the increase in breast cancer rate, there's a little blue line that runs across the bottom. That's male breast cancer. If you expand male breast cancer, the interesting thing is that the rate of male breast cancer has stabilized at a point 20% higher than where it was in the 70s. Men do not take hormone replacement therapy. Men do not have mammograms. They do not have a standardized screening program. You have a 30% increase in the incidence of male breast cancer, and I didn't put it on the slide, but Dahl and Pito also observed that there was about a 20% increase between the 1940s and the 1970s. You can't explain women and blame it on a whole series of other things unless you can explain men, and you can't use those factors to explain men because men don't get mammograms, et cetera, et cetera. So there really is something going on here. So as we think about going forward, we do need better epidemiology. Now, this paper, many of you probably have seen. Actually, somebody here, and I can't remember who it is, recently improved on this with a study that was just was actually done here at NIEHS. I can't remember who the first author was. Of somebody, pardon? Okay, but this has been improved on. But this is the one I found out about this article when a friend of mine sent me a link to a blog saying Goodson is crazy, because I had been involved in a paper looking at bisphenol A. And I looked at this and I thought, holy crap, you know, this stuff goes in and comes out. And this is done by giving people fruit cocktail and looking at what happens. You can get kind of the same kind of thing, I think, by giving people canned soup versus homemade soup. And you get a spike and it goes up and it comes back down. And I thought to myself, wait a minute, you know, maybe there is something, maybe we've made a mistake. But then I put on I started out to be a pediatrician. I put on my pediatric hat, and I remember pediatric endocrinology, and I remember people talking about spikes. And so what I did is I started looking into it and said, yeah, I did remember right. Spikes are the way that normal endocrine hormonally driven physiology works. Nobody gets a little bit of an increase in their LH, and I can't remember which one, in one of these, this is the LH level. Nobody gets a little bit of increase in LH and then has a little bit of increase in their estradiol as a level thing. Biology works by a spike, a response, a spike, a response, a spike, a response. So if somebody's sitting there dumping spikes into somebody, if they have fruit cocktail twice a day, I think that's very logical, that could be reproducing the normal pattern. And in fact, this is what the normal pattern is. And if you do this by Tanner scale, this is age 6, age 10, 9, it's a little blurry, and this is about age 15. But Tanner scale is a way of categorizing breast development, pubic hair development in young girls. And the key thing is that red line is sleepy time. Everybody gets a little bit of a spike in their hormones at bedtime, but early on, you don't get spikes when you're just starting to develop. It's only as you really start to get breast growth that you get spikes. And when you get spikes later on, when you're really getting breast growth, you got a crazy spikes. It's not a difference you're running. It's a question of whether it's blip, 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 or blip, 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 blip. And so I think one of the important things, one of the really important challenges in all of this is not going to be just sitting there and figuring out what chemical dropped on what will do things. We need to have a better understanding of the physiology and how this interacts with the physiology. And the other thing I always point out is that diethylstilbestrol also clears very, very rapidly. We don't accumulate DES. DES is gone within about 18 hours, all functional purposes. The next slide is simply to point out that if we don't want to be focused on xenoestrogens, because when we focus on xenoestrogens, we tend to look at the cells, we tend to look at how they grow, and that's not necessarily exactly the only way things can happen. This is actually a study from our lab, and what we've looked at is how exposure to a chemical will interfere with tamoxifen-induced apoptosis. And so what you can see, this would 100% would be up here, and you suppress apoptosis, and this is 3 cell lines that have developed out of our work, plus T47D and MDA231, which is an ER-negative tumor. The MDA231 doesn't make much difference, but all of the other cell lines, you do get a suppression of apoptosis after exposure to terephthalic acid. The interesting thing is that you get a suppression of apoptosis, but you don't get stimulation of growth. This shows the shift into G1 arrest. These cells go so far and they have a G1 arrest that's associated with P53, a marked increase in P53. And then if you start saying this, okay, why is this cell evading apoptosis, which is what you get with BPA, but is stopping in arrest, what you find is that there are all sorts of markers of DNA damage and DNA repair that kind of go through the ceiling. And the interesting thing about this is if you then take away the terephthalic acid, all of this reverts, you continue to have the evading of apoptosis, but all of this reverts, the cells take off and grow like crazy, but the markers of DNA damage persist. So what you really have is a situation where a chemical will induce DNA damage, and at the same time, when you sort of take the chemical away, the cells then continue to grow with the DNA damage. So focusing just on, if you were doing a screen test looking at S phase as your principal test, this would be a safe chemical. I don't think it is, but I think we really don't know. The next challenge is to find good models. Cell line unreliability or contamination is a repeated topic. Nobody wants to deal with it. MCF7 is you don't quite know what you're getting. Burrell talked about this 5 years ago, recent article in Science, quoting Korch, talking about the variability in the cells that aren't what you think they are, and the ubiquity of the Henrietta Lacks' cells getting into everybody's lab. We have sort of taken the viewpoint of trying to study carcinogenesis in non-malignant cells. It can be done. The tox people are growing cells from various sources. We grow cells from fine needle aspirations. You can get the sample, you grow it in the laboratory, you can grow it without or with various chemicals. If you grow them in gel, they do what they're supposed to do. And so that's one part of it. And the other part of it, and what's going to be a really big challenge in all of this, is to figure out how we're going to tie this into people. Because we can look at individual results in the laboratory, but we need to start thinking through what are going to be real tests that we can do in people. This is really complicated because it's hard to find a control group for anything. There are some studies done with intentional exposure to BPA, the one that was recently done here. They're the cash register receipt types of studies, and those are one thing. The other possibility, and we've started working with this, is to work on withdrawal of exposure. Identify people who use a whole lot of stuff and try to provide them with alternative products. And this is a bit of a logistical nightmare, but at least there are really two nightmares. One is getting people to do it, A, recruiting, B, getting them to do it, and C, what can you expect to revert in the course of a month? And trying to get somebody to do this for more than a month, I think, is going to be really hard. So it raises some really difficult questions that have to be addressed. The next thing we need to look at is we do need to start looking at mixtures, as was sort of the basis of this. This is Philippa Darbre, and I give her a lot of points because I think she's done one of the most interesting studies. What she did is she got mastectomy specimens, worked with a analytical chemical group, and measured the levels of different parabens in the mastectomy specimen. So she knew what was in the tissue at the time they had cancer: methyl, ethyl, N-propyl, isobutyl, butyl, N-butyl, benzyl paraben, all this sort of stuff. And then what she did is she exposed, for all the problems with MDF7 I just talked about, she exposed MCF7 cells to these parabens in the same concentration that they existed in the subject. And what she found was that quite frequently you would have sort of minor changes, but if you reconstituted the cocktail, you had a very significant increase in cell proliferation. Same cells, none of them individually, and the mixture. So the mixture that exists in humans has been shown, at least in that setting, to actually work synergistically and actually using levels that were actually measured in human tissue. We've taken a slightly different approach, or not a different approach, but a continuing approach, and looking at dissimilar structures. We've looked at bisphenol A, methylparaben, and PFOA in what seem to be environmentally relevant concentrations. And we've started, this is just beginning work, but I point it out because it points out a couple of things. We've looked at S phase and what it does to S phase, and we've looked at apoptosis suppression. And what's interesting here is that you see two things. And one is that when you look at the level of S phase that you get, that the mixture always is a little better than any one of them individually. if you look at the mixture with the T47D, the mixture is much more impressive than it is in the MCF7 message. Whatever cells you choose are going to influence your results. So you've got to be really careful about the cells you choose. And the same thing is when you look at suppression of tamoxifen-induced apoptosis, MCF7, the mixture is more effective, and actually it's slightly more effective than estradiol. And when you look at the suppression with in T47D, you also get a lot of suppression. But you also can compare what you get with bisphenol A here and bisphenol A here by the cell line, or look at what you get to S phase by the cell line. The cell lines that you're using make a huge difference in what's going to happen. So finally, we get around again to this question of epigenetics, and epigenetics is going to be the headache that all of us have to deal with, because in terms of understanding how cancer happens and how it persists, the persistence of epigenetic change is going to be this little thing that we have to account for. And as this is from actually Dow Chemical, LeBaron and several people put out a paper and they said, without detailed characterization of the epigenome, including elucidation of the stable variable methylation, it'd be difficult, if not impossible, to determine if an exposure to an exogenous agent that leads to an he's very wordy, epigenetic alteration. It's a deleterious effect as whether part of normal epigenetic shift in variability. That's absolutely true. It is a challenge that we need to deal with. And at the same time, it spells out the fact that if we solve that, we have solved a major issue. So go read the epigenetic literature and then try to go to sleep at night. This is a study that was done with Inuit, and they have a fairly high exposure to organic chemicals. You could show a slight decrease. The ALU measures a larger number of methylation sites than the line one. Line one had a trend. ALU was actually reached statistical significance. And what you could see is that it was sort of you had hypomethylation associated with chemicals, and you turn around in elderly people and you looked at looking at specific genes, and you found hypermethylation of the aryl hydrate receptor gene. So you're sort of like, what do you do with it? And then you start getting into it. And these are the first two there. And then Ito et al., they looked at women, and they found that but they were looking at leukocytes and inverse methylation. So you had more organochlorines, you had less methylation. This one is looking at maternal DDT, and again found an inverse relationship in whole blood methylation. And then you have this one, which looked at DES, and there was no association of methylation between women who were exposed to DES or not, but they're looking at methylation in leukocytes. And the reality is that we have to come up with a way to actually look at the target tissue, which is either going to be breast tissue or the vaginal tissue. So in both these situations, we need to be able to sort out what methylation issues are, but we also need to figure out the clinical problem, and this has been something I've worked with years. How do you get samples from people to actually look at DNA methylation in the breast tissue? And that's a really tricky question, but I think that's something that as we try to figure out how this relates to people, it's either going to be epidemiology or it's going to be samples where we figure out how to sample people. So as I said, the challenges are better epidemiology. We need to understand how this interacts with physiology, and you need to look beyond simply xenoestrogens. We need better models and ways to use human studies, and it's obviously the whole message of the thing. We need to look into the combination of dissimilar structures and functions, and we need to understand chemicals and epigenetics. So as I thought about the ship, what makes the cruise industry so successful is that the cruise industry they have wonderful radar and satellite tracking and everything like that. If there's a storm, they just go someplace else, and they're fast and they can get away with it. So that the cruise ship experience for the passenger was always good. But when you sail out into the North Atlantic, if you really just sail out in the North Atlantic where you're buffeted in all sorts of directions, you can't really get away from things. And so part of the challenge for us is to kind of start looking at what the perfect storm might be in terms of what would happen to our patients. Thank you.
Okay, actually, why don't we invite both of our speakers back up and we can address questions around this topic, the history of the Halifax program, how we got to where we are, and some of the data slides that we've just seen a whole bunch of. Do we have any questions? First, let's go to our webcast folks. If you'd like to submit questions, we should be posting right now on the webcast the address, the email address to which you can submit those. And then we'll go to folks in the room. If you can come up to one of the microphones in the front so that everybody on the webcast can hear you as well.
What do we know about the sequencing of the various steps in the biological process leading to cancer? Is it a decathlon where you have to do all of the steps in order, or can you do one here and one there?
I'm afraid that's really part of the challenge that we posed, is that process is not well understood. We've got examples of ways in which cancers have been initiated, but, you know, I'll give you an example. If you think about the way we started doing testing with two-stage, with damage and promotion, if you could get cells proliferating, it's my understanding that once you get cells into a tumor, you create this sort of hypoxic environment. There is the ability for them to sort of, over time, create the right mutations necessary through the genetic instability in that environment to be able to generate subpopulations that have all of those needed aspects. But in terms of, is it a decathlon, one or another, or some different order, that's really why I put a black box on my last slide. And a lot of the teams in the project complained that there wasn't literature on the process to help them better understand, if we're going to talk about what does it take to get cancer underway, do we only need few hallmarks? Do we need many? Do they, that's not well understood. And there's a few papers on that question, the fact that, or that lacuna in the literature, that there's not good work on that. So that's my understanding. Nobody from the project came to me and said, oh, we need to share this resource. Everybody came and said, we're having a hard time finding information on what carcinogenesis looks like. I think we raced ahead after being at that sort of early stages of chemical carcinogenesis and producing chemicals. The tobacco industry was doing co-carcinogens 30, 40 years ago. They understood the synergy thing, but then everything shifted towards therapy and biology. And, we have enormous effort at the NCI to do therapy and biology, but the causation part is really not there, and that's a piece that we need to go back and revisit. I hope that we would highlight that in this project. Sorry.
I think the other part of that is that genetic mutations do tell us something, and that is that you can start with a genetic mutation, you can start with the absence of P10, and you can start with P53 mutations, all of them end with cancer. So obviously you can start sort of at the beginning, the middle, and the end, and end up with cancer. So there may be things that have to happen in sequence, but biologically we know that you start with a problem at the end, everything else will fall in line.
So I have kind of two questions. The first one, Bill, really has to ask you of how important you think it is to know what the ratio of chemicals are in someone with cancer, when in fact that cancer started a long time ago. Especially you showed Barbara Cohn's data from the Children's Environmental Development Program, I think is what the California study has been called,
Right.
which clearly shows that the windows of susceptibility are early in life, at least for DDT exposure. So I'm often perplexed by people who spend a lot of time looking at what's in a tumor. It's fine if you want to look for mutational signatures, so maybe you can develop precision medicine and therapy, but I think, that horse left the barn a long time ago.
I think, unfortunately for the patients, the figurative horse has usually left the barn a long time ago. There have been some studies, actually were done at UC Davis, I think it was, where they took fat tissue adjacent to cancers and fat tissue adjacent to benign lumps, and they were looking for differences. They couldn't find differences. And I think that a lot of what goes on is looking at where somebody is now is not going to really solve where they came from. And one of the reasons I pointed out that we're not looking at things that are yes or no phenomenon. We're looking at things that are a bit more or a bit less. And it's to the degree to which we're causing more cancer that we need to worry about it. But it also means that there are huge numbers of people that are going to be out there who've been exposed to the same things, and for whatever reason, it never quite crossed the line. This was the same problem with lead water pipes. 100 years ago, people would argue lead water pipes were safe because they would show households where nobody was sick, and then they'd show, somebody would say, well, the households where somebody, everybody was sick. And you can always find people with the same exposures who are not affected. And so measuring what there is right now may not really be the right approach.
I just like to make one comment. I always like to think about smoking. Smoking is one that perplexed us for a long time because people had this sort of obvious mixture exposure, and it could happen a long time ago. But what I find is remarkable is that something like 2/3 of people who smoke don't get cancer.
Only 11% ever get lung cancer.
Okay, so there are big numbers of people that don't get cancer, and yet we know all of the seemingly right constituents were there. And I think part of this is when you think about some of the genetic defects that Bill talked about, they create risk levels. It's like prone to. Some percentage are going to get this, but some aren't. And so now we have to think about, well, if we have some subpopulations within the general population that are prone for various reasons that may have occurred many, many years prior, what does it take to put them over the line? What does it take to develop that possibility into a cancer? And it may be that the kinds of chemical disruptions that we're talking about is enough, not necessarily to act on all the hallmarks, but acting on certain phenotypical aspects of cellular biology. We then take somebody who is at risk and may never have developed cancer into somebody that now is going to turn it into a cancer.
I totally agree with that. The other issue I wanted to raise is, Bill, you're calling for the need to do epigenetic studies in the tissue of interest. And I agree with you up to a point, but most people are not interested in having you take a sample of their brain or their kidney or something else like that unless they have a major problem. And so that I think this is an opportunity for toxicologists to work with epidemiologists and clinicians and develop the relationship between the epigenetic marks that might occur in a target tissue and the epigenetic marks that are available in accessible body fluids, and then kind of use the parallelogram approach to go into humans.
I want to add one other thing about humans, and Bill and I had a quick discussion on this last night. The question of how do you validate things that we can demonstrate in vitro or in vivo, in rodents, et cetera. How do we bring that to the human population? Because we can't study in humans, obviously. One of the things that I think we didn't really address in this project, but I think there's a future for perhaps, is the molecular epidemiology. Once we have a better understanding of how that unfolding of carcinogenesis occurs, we could revisit big data sets where we have exposure with large populations of people where we were looking for single correlations of single chemicals historically and single cancer types. We actually could look at combinations of chemicals and have justifications for why grouping those chemicals makes sense. And then we can look at how cohorts behave, and that gives you more statistical power, and I think that might help us as well.
I think the only other comment I would add is, in a general sense, I don't think people are going to let us biopsy their brains. I do agree with that. On the other hand, we have found that women will let us do breast biopsies, needle biopsies, and on a volunteer basis. And so it's, the first time I went to the IRB, it was kind of like, what the heck are you doing? But I think that part of the challenge is that we really do need to think about how we're going to do those kinds of studies and what will be palatable to people.
So we have a question from the webinar. This one comes from Neetu Singh. And she says, it's true that we're exposed to environmental chemicals which may lead to carcinogenesis, but I also suppose we need to check the balance of antioxidants present in the diet, which balances the exposure of these chemicals.
I think that it was alluded to earlier, there are dietary changes. I think the blanket assumption that antioxidants are good for us is not supported by the experimental studies. I mean, people have looked at I think it's lung cancer, and it doesn't necessarily go down in people who are on high doses of antioxidants. So I think that I always point out that we always think about reactive oxygen species as damaging DNA, but you have to have an elevation of reactive oxygen species to trigger apoptosis in the treatment of cancer. So if you truly can get rid of all reactive oxygen species, you're never going to get apoptosis, and apoptosis is the basis for chemotherapy, radiation therapy, and hormone treatment. So it's something we need to sort of understand better. I would put that in understanding the physiology better.
Over here.
Yeah.
So just wondering, in your analysis, did you actually address the interplay between some of these hallmarks of cancer? So for instance, with DNA repair, an increase in MBS1 will affect the epithelial mesenchymal transition. So I just wondered if you were able to capture any of that.
There's 10 teams that did individual research on individual hallmarks, and all of them were asked to address the interplay between the mechanisms in the hallmarks. Of course, that's a very big task for the teams and what can be accomplished in an individual review. And if you look at the individual reviews in the special issue, say you can pick angiogenesis, for example, one of the things they're supposed to answer in the review is, what do we know about the way in which other hallmark phenotypical mechanisms are related to angiogenesis, and they try and do that in a succinct manner in each paper. So there is a drill down there. I will only say that, it's a lot. If you look at the breadth, these reviews were atypical in the sense that the breadth of the reviews were substantial, and they had a lot to accomplish in 30 pages or whatever they had. So there were editorial decisions that had to be made about how much do we focus on that. Some teams did a better job of that than others, but I would encourage you, if you're interested in the interplay between those mechanisms in the various phenotypes, to go to the individual underlying reviews that led to the capstone. We really started with 10 reviews of each area, and then we built the capstone from that, which was the Goodson et al. Paper that we've been referring to.
And the other part of it is you identify a big hole in our knowledge. I mean, you are pointing to something that we're beginning to sort of think about. But, you identify, your question is excellent, and we couldn't have asked for a better person in the audience to ask it, because it really identifies the challenge that we think needs to be structured into how we think about, what's toxic and what's not toxic, because we tend to think in terms of single processes rather than spillover processes.
Hi, Kim Lyerly from Duke. Again, I want to say congratulations to Leroy and Bill and Margaret. Really enjoyed your comments. One of the things that I think you're not really mentioning is the whole social dynamics of creating a group of nearly 200 scientists to come together and develop a consensus. And again, I think there are many lessons to be learned from the context of the reports. But I also think that that capacity to bring people together and to try to work on a common theme in a way that's productive is something that really should be underscored, and maybe that's an important lesson, because we may not address all the questions today, but certainly that capacity to pull people together is quite remarkable. And then the second comment I wanted to make is that you did pull in a bunch of individuals, and at Duke we have a School of the Environment, and we try to build bridges across those types of schools. Some of the comparative biology is to look at other species and so forth. But one of the questions I had was, there are a lot of people who have an interest in canine breast cancer, and there's a big incidence of canine breast cancer and so forth. Do you see the same type of trends? And Bill, would that be a model for environmental exposures that you think would be relevant and potentially useful in future studies?
I think it's a fascinating question. I don't think it has an answer. I think it's beagles that really get breast cancer. And I actually I went and talked to a veterinarian one time about whether we could, because he does a lot of actually breast surgery at the vet school at Davis, and he said, well, this is all very interesting in your chemicals, but this is totally off target. He said, what I'm really concerned about is the amount of amount of progesterone that everybody gets all the time. And I said, what are you talking about? And he said, all of your dairy products comes from pregnant cows, and all of your dairy products are full of progesterone. And in fact, we did the study where we had a bunch of guys come in, eat dairy products for 24 hours, and you can drive their progesterone level up. And his comment was, it is much broader than what we think. On the dynamics of getting a group together, I think that my hat was off to Leroy. My hat is off to Leroy. He did a fabulous job in getting all of these people together. And I think one of the things that I was going to mention on the slide that where I was mentioned in a blog as being a blithering idiot because I didn't realize it was gone so quickly, is that this is not something that we should not be doing studies by ambush. We should be talking about things. The epidemiologists and the toxicologists and the biologists and the oncologists, they all need to be talking to industry, and there needs to be a back and forth. This whole thing about, well, it can't be this because, it goes in and comes out, and so like anybody says, well, excuse me, but that's how biology works, is something that we need to sort of step beyond this, because this is not really something that's the stakeholders here are not the doctors and industry. The stakeholders are our children, our grandchildren, our great-grandchildren, and whatever we do now is going to change their lives, and we really, it behooves us to try to do it right.
I just want to respond to your question about the veterinary aspect. Well, one of the scientists in the project was Elizabeth Ryan from the University of Colorado. She's at one of the animal cancer centers. This is a whole network I was unaware of previously. They have 100 or so cancer researchers dedicated to veterinary oncology at University of Colorado, and they're one of a number of centers around the nation that collaborate. And anybody who has therapeutics, for example, and they're looking for 100 dogs that they need of a particular cancer type, they have a database of what's available at any one time. Parents can bring their pets in for treatment and et cetera. Elizabeth was on both sides of she was on two teams on both task forces, the therapeutic side and the environmental mixtures work research side. And she's been doing research specifically on chemicals that dogs have been exposed to and what those combination effects look like and how they compare to the kinds of cancer she's seeing. And I would encourage you to reach out to her. There is a whole network in place that are already collaborating for therapeutics, and I know she's one example of somebody who's specifically involved in the environmental health side of things, so I think there's probably others.
Could I expand on that, please? I'm over here. The Canine Lifetime Health Study is being run by Morris Animal Foundation out of Colorado State. Rod Page is the director of that project. They have 3,500 golden retrievers that they're studying over their lifetime, from young 6-week-olds until they die, for cancer specifically. But they're really looking at environmental reasons for the health outcomes that they have. And it's not just breast cancer, it's any kind of cancer, any kind of health adversity. So they're looking for people in epidemiology and the basic sciences to collaborate with them. They would love to answer some of the questions that are being raised, especially because they're looking specifically for environmental, chemical factors that may be involved.
So you're touching on three very difficult areas here. Looking at low dose, you're looking at chemical mixtures, and you're looking at cancer. Why cancer? I mean, if you looked at low dose, you can look at much more immediate endpoints. If you look at chemical mixtures, you can look at much more immediate endpoints. So what is the reason to combine these of the most difficult problems into one?
Well, you'd have to look at the genesis of the project. I was sat by my grandfather's bedside when I was 18 for a couple of months as he was dying, which left me moved deeply by what cancer patient looks like. And later in life, his daughter, who was my aunt, who was a childhood play friend, she died in her 40s, and I got interested in cancer. And I spent my time learning about the disease and reading the peer-reviewed literature, and I started pulling things together. And it was really trying to understand, what do we know about the environment in cancer? And the endocrine disruption work that was happening at the time was timely. It was interesting. It was relevant to the challenge. What was happening in traditional toxicology was interesting. What was happening in cancer research was interesting, and I tried to say, well, what does this tell us about the problem? was far beyond my own ability. I mean, conceptually, I had the broad strokes when we started the project, but when I brought the researchers together and said, this is what we wanted to tackle, a lot of them said that makes a lot of sense from their understanding of the biology and what they think is going on and the issues. They thought it needed some attention. And I won't deny that it's a challenging problem, but I think, we've got a lot of bright people and a lot of good tools, and, things are moving quickly. I think the timing is good. That's why. There are easier problems out there, I'm sure.
First, thanks to all three of you for just brilliant talks and comments. Very, very stimulating. I was wondering, Bill, if you could comment on the problems that you and others have highlighted with using different cell lines, and whether that really brings to bear the need for things like primary human cells and stem cells and complex 3D organotypic culture models of tumorigenesis, both potentially from healthy patients or from cancer patients in pre-metastatic or pre-neoplastic scenarios, and whether those might be very good platforms for testing the toxicity of certain chemicals and interrogating chemical effects, mixture effects on different hallmarks and different phenotypic changes, or whether you see genetic heterogeneity as just too much of a confounding factor there.
Two parts to the answer. The first part is that I started out studying wound healing, and I really know a lot about wound healing in Sprague-Dawley mice and C57BL Jackson mice. And along the way, I got interested in how you can test healing in humans, human diabetics, and found that the results in humans were not the same as the results in the mice or the rats. And that was kind of a real eye-opener. So I think it's extremely important to figure out ways to test humans. I did studies looking at cancer growth rates measured in humans in vivo in labeled BUDR, and I wouldn't do that now. But this was something that seemed like a good idea 30 years ago. And so I've done a lot of work with humans. And I think what I find, and my very strong opinion, is that the human and the human variability is very important. When Venter and the other group put out their genomes, what everybody said, we know the human genome. We didn't know the human genome. We knew Venter's genome. And that's a difference. And one of the things that we've done by growing cells from fine needle aspirations is that our profile on bisphenol A is not based on a cell line. It's not based on one person. It's based on results that are concordant results from about 60 people. And it's being able to show something across a large number. So my personal prejudice is that cell lines are very interesting for working things out. And I would not detract from what you can learn from cell lines. But when you try to figure out what really happens in people, you ultimately have to go study people. We had a joke in medical school, we'd study E. coli more, or we study people so we study people so we will have a better understanding of E. coli. And this is really the way we ought to be thinking. We should be thinking that we need to figure out ways to study people. And I recognize that people are not going to want brain biopsies, but I think that we started out doing breast biopsies with a needle under anesthetic. When somebody was getting cancer on one side, we look at the cancer cell, look at the benign cells from the other side. We check that they're benign. We've done SNPs. We can prove they're from different people. It's not some sort of lab contamination. It can be done, and we've moved into doing it on people who are basically awake and chatting with me. It's amazing what you can do with local. And so I think that part of the challenge is the imagination to figure out how to do that. And I think that's going to be an important concluding step in all of this. And I think repeated fresh sources. The other thing is I think that a lot of what we do is with cells that we don't assume our cells are going to grow forever, because normal cells don't grow forever. And if you have a cell that grows forever, it ain't a normal cell. But you can look at the ups and downs and the perturbations of things when they're fresh cells. So renewable cells, I think, are extremely important. They're also a pain in the neck. Seriously. I'm always doing the IRB, and you got the 13-page consent, and it's like one at a time, and it's a little, it's hard work. And if you're going to do it in a lab as a PhD, you've got to find somebody who has access to clinical tissue who wants to work with you. But, you know, there are people at University of Kansas, there are people at Northwestern, I'm in San Francisco, who are working with this. It's not that hard. It's a question of involving clinicians to get your source material, and that can be done.
Okay, so I'd like to thank our speakers again. They did a great job this morning. And their work, I guess, in some ways is done now, and it really falls on the rest of you. We have a great group of folks here from government, from NGOs, from academia, and from industry, and I think each of those brings a unique perspective to help begin addressing some of the questions that we've seen. A lot of the research that we've read about in Dr. Goodson's paper is really asking more questions than it's answering. And a lot of those questions we're really going to define and work through this afternoon. And that's where this isn't a spectator sport. This is an opportunity for each of you to really contribute to this effort and to the directions that research goes in the future. Just because of time, I'm not going to go through each of the individual roundtable charges, but suffice to say, we're looking at it from a past, present, and future direction. We're looking for, in the first session, information gaps. We look at what has been done and figure where the gaps are, what types of research and what areas can we really make large leaps forward as opposed to small and incremental changes. The second, with research methodology, is really a present. What do we have as the tools to be able to begin to address some of those questions that we're now asking? And then finally, the future is how we can apply some of these low-dose mixture theory concepts in cancer to non-cancer and to policy and decision-making and the way in which we live our lives to have better outcomes. So I encourage you all to take some time now for lunch and then come back. This room will be divided up into 3 separate areas that we can have discussions and debates and really bring up some of these topics and issues of concern. And then we'll get back together. I believe it's at 2:30, and we'll be able to hear a report out, actually 2:45. We'll be able to hear a report out from these groups in order to see the way in which each of these groups' information is actually interacting with each other. We're going to extend lunch. We'll be back here at 12:45 to start with the working groups.